Thank you, Roy. Does everyone have a handout? Does everyone not, I should have asked, does everyone not have a handout? All right, so this is a quiz. No, just kidding. So we are going to, who was in charge of this? God. Can you hand one back? So we're going to cover a lot of ground, and a lot of the topics that we're going to cover are on here. A little short description of what we talked about, and then a QR code so you can start your own kind of rabbit hole looking at, hang on, I can't do two things at once. All right. Yeah, so you can start your own rabbit hole of digging into some specific topic, because what we're talking about here today might be totally new to you, but after you hear it once, you'll probably hear it a bunch on the news, on the pickleball court, somewhere, and so you can kind of dig in and say, oh, I remember Ryan said something about neural networks. What is that? You can come here, and that be your starting point from which you can kind of figure out new things. All right, so I'm really excited to talk today about how we are trying to put patients at the forefront of clinical decisions. That might kind of sound weird, like where else would the patient be when the doctor's deciding how to treat you? But your doctor right now probably already knows for almost any condition what medication they're going to use and at what dosage before you even walk in the door. And I want you to look around. What are the chances that all of us need the same medicine at the same dosage, right? It's probably pretty low. Now, I don't mean to knock your doctor. That's just the data that we give them. So what we want to do is we want to figure out the best treatment for you based on your genome, all right? And to do that, we are going to talk about how we have these really large data sets that have medical records from hundreds of thousands of individuals. Think of those medical records as little journeys that people went on in figuring out what medicine at what dosage worked for them. We want to find the people in that big data set that look like you, and then figure out from that matched cohort how the doctor should treat you, all right? And so to do that, we're going to talk about how drugs are approved. We're also going to talk about machine learning. So quick question, who has heard of and knows what artificial intelligence is? All right, who's heard of machine learning? Who knows what the difference between those two things are? So I'm going to try this. Doing live demos is always the worst thing to do in the world. And so to get the answer to this question, I want to just ask straight from the source. So has everyone heard of ChatGPT? So if you haven't, this is this really, really impressive technology called a chatbot. And what you can do is you can basically interact with this thing like it's a person. So I'm going to ask it, what is the difference, I can't spell, but it's okay, it figures it out, between AI and machine learning? Okay, well, that's a lot. Oh, it's talking. All right, so let's make that really short for me. Okay, so AI is the broad field of creating intelligence. Why does it keep talking? And machine learning is how we get there. Machine learning is a set of algorithms that will help give us artificial intelligence. So just from now on, in popular media, those two things are overlapping. AI sounds a lot more exciting than machine learning. But the way that we're going to get to AI is through machine learning. Machine learning are computer programs that take really big data sets and figure out patterns within them to answer these complicated questions, like this one in front of me that just takes natural language and spits natural language out. What it did is it trained on basically the entire internet and just kind of guesses based on, make that really short. It has seen that enough and seen the results of that enough that it can guess how to change its old answer to its new answer. Now, it's important to know there's a lot of ethical problems with this. I have a lot of colleagues who think that ChatGPT stole their work and trained on it and is now putting it back out. I can actually get this thing to give me copyrighted images back, which is a big problem. But that's kind of far field. It's a really exciting technology that will change how we teach and how we do medicine. All right. So anyway, back to this. So we're going to talk about big data sets. We're going to talk about machine learning. We're talking about how drugs are approved. But we're going to start at the very beginning of all this about genetics. And the biggest question, what is a genome? It's a pretty big question. Has lots and lots of answers. If you Googled this, you might find something like this. We have trillions of cells. Our cells have nucleus. Inside the nucleus is our genome. Yada, yada, yada. That's right, but I don't think it's that insightful. The definition that I like is your genome are the instructions for your life, which is a pretty big answer to a big question. Seems a little bit abstract. So to figure out what that actually means, I like to think about milk. That's kind of random, right? But it's just essential for life. It was your first meal and probably your only meal for months. And if you look at this picture and you think, I could really go for a cold glass of milk, or you think, get this off of the screen, it's because you have specific instructions that tell your body to act that way. All right, so what is milk? Milk is a lot of things. The primary carbohydrate in milk is lactose, which is two simple sugars, galactose and glucose, bonded together. So when you take a big swig of milk, you're basically flooding your intestine with lactose. And interestingly, your intestine can't do anything with lactose by itself. And if it sits there long enough, bacteria will start to digest the lactose and give off all sorts of nasty byproducts, including methane. And you either can imagine this or might know this directly. That's not pleasant for anybody. But thankfully, the human genome on chromosome 2 has the LCT gene. And in the scope of life, think of a gene as a little instruction booklet for a little corner of life. So these instructions are in the form of DNA, and they are instructions to build a protein. And the way we read these instructions is 3 positions at a time. This is called a codon. It's just a weird word, but that's the one we use. And the first codon is a special one. It says, it's a start codon. Start making a protein, okay? And then we're gonna move on to the next 3, and what a protein is a giant string of amino acids. So, not surprisingly, the instructions say, well, the first one's an E, and the second one is an L, then an S, and one by one, we'll have the instructions for, and we'll add a amino acid to this growing protein. We'll slide all the way down to the end of the gene until we reach another special codon that says stop. At that point, most of us will have a lactase enzyme. And the whole point of this one is to break that bond, leaving us just galactose and glucose. So instead of a bunch of bacteria, we have these simple sugars which our gut can readily absorb. Now I say most of us because some people actually have a slightly different version of this gene. So if we slide about a quarter of the way through, this little T there in a few people in the world is actually an A. Big deal, right? Yeah, big deal. A TAA doesn't encode a Y, it encodes a stop. So think you're making bread, you get a quarter of the way through the instructions and you slam the instructions, you've got to eat that. Well, that's not a loaf. And this does not produce functional lactase. So whereas most people have this lactase production curve where it ramps up before birth, stays high until you wean, and then tapers off, these individuals don't produce any lactase. They have something called alactasia. Alactasia, and as you can imagine, is a pretty serious condition for infants. They have to be taken off of mother's milk and given lactose-free formula, which isn't great, but it gives them the nutrients they need to survive. So you might be looking at this graph and thinking, I weaned a long time ago. I still like milk. That's because you have slightly different instructions than most people on Earth. You have something called lactase persistence. And the instructions for that specific version are actually outside the gene and they keep the gene on for your entire life. And you'll probably have a modification in one of these 3 areas. And interestingly, these 3 modifications have a known origin. They either come from, the 2 Gs come from Eastern African pastoral societies, and the red one comes from a European society about 10,000 years ago. The idea here is these people had domesticated cows. They started drinking milk. This mutation was already in the population, and those people that could digest milk as adults did much better. They had an extra source of nutrients. They could convert grass to calories in a way no other human could, had bigger families, and now about 40% of the world's population has these modifications. So the idea is that your genome, your instructions, 1, are a product of your ancestry. 2, they affect what diseases you're susceptible to. And for us today, we're going to talk about how they determine what treatments will be most effective. And the classic example for this last point is warfarin. Very, very, very common medication. Came about in the 1950s. Interestingly, it was previous to that, it was a rat poison, which is bizarre. But getting the dose of warfarin, which is basically a blood thinner, right, is super important. Too few, too little, and you have no effect. Too much, and you bleed out. And the dosage is so important, in fact, the manufacturer comes up with all these different colors that correspond to concentrations to make it a lot easier to get your dose just right, so you don't mess it up. So you don't have to remember you need 4 milligrams, you remember you got the blue one, all right? While this is essential medication, it's also one of the top adverse drug reactions that we know of. So we've been going along with this essential medication for quite a long time. In 2013, we actually realized that not everybody reacts to warfarin the same. In particular, people of African descent need slightly more, and people of Asian descent need slightly less. So you can imagine if we have one regime for figuring out how much warfarin you get, and we don't take into account your ancestry or your genome, we're going to, for a lot of the world, be giving the wrong dosages of, for example, warfarin. And you might imagine, I don't have the numbers for this, but I'm imagining many of these adverse drug events are because those connections weren't known and weren't followed. So you might ask, well, how the hell did we get here? Don't we test these things? Yeah, we do, but we test them on really small populations relatively to human population. And then we look at however the average effect was in that small population, and that's what we give it to doctors. We say, okay, this is a safe, effective dosage, go forth and now prescribe this to your patients. And the way that we find those dosages is what's called double-blind randomized controlled trials. Now, this is a mouthful and probably something you didn't care at all about maybe way back in 2019, right? But we all probably followed the clinical trials of the COVID vaccines very closely. Like, I personally remember where I was when I learned that the Pfizer vaccine was 90% effective and safe. I was in Estes Park after my sister-in-law's wedding. It was 3 o'clock in the morning, and I was driving my infant around Estes Park because he wouldn't stop screaming. And I heard that on the radio. It was just amazing news. But so you probably have your own little memory of that. It was an amazing outcome. And it followed, and most other clinical trials have this as one of the final steps before being released into wider patient usage. So here's the overall protocol. We'll follow it. So you start off with your study population. So in the case of COVID, it was just a whole bunch of volunteers. More likely, it's everybody in a hospital system. So this is the people you have to choose from. Now, there's a inclusion criteria step that happens. Inclusion criteria are all the people who you think might benefit from your medicine. They're the right age, they had the right diagnosis. Now, there's also exclusion. You don't want to hurt anybody. So anybody that has some sort of medical history that might put them at risk, we exclude them. So we have a big group, we exclude and include people, we end up with our study sample. Now here's the random part. We take that study sample and we sort them into 2 groups, either the intervention group or the control group. And double blind, no one knows what group they're in. The doctors don't even know what group you're in, all right? So you're in a group, the intervention group gets the drug, the control group gets the placebo. After some interval, we then count who got better and who didn't get better in each group. And we do some pretty basic math. We take these numbers, we put them in what's called a contingency table, and we run some statistics on this, and we ask the question, are the number of people that got better with the intervention statistically significant? And in this case, this is really small, obviously, but in this case, yeah, it is statistically significant and we'd approve it. To give you a sense of how conservative the math is here, if I futz with these numbers just a little bit, right, you can still see the pattern, right? It looks like if you got the intervention, you're more likely to get better. But in fact, even at this small change, this is no longer statistically significant, and this would be rejected. And actually, most drugs get rejected in clinical trials because they don't have as large of an effect as we anticipated in humans. All right, but let's say it's approved. Well, what does that look like to your doctor? Well, it's in fact just a gigantic PDF that talks about adverse effects, but really, this is the table that's most important to them. This is the initial dosages. So you can see for this blood pressure medication, 2.5 milligrams is the low ball we might start somebody at. But there's this really interesting, maybe troubling sentence before that. Those doses may be titrated upward, that means increased, until 2 things happen. Either blood pressure is controlled or you reach 40 milligrams. There are people who started at 2.5, went all the way to 40, and their blood pressure was not controlled. That sounds painful, right? And we think the reason that there is this big disconnect between these dosages and what you need is because treating people one size fits all, let's take the average effect of this trial and make that how we start you, doesn't really work, right? So let's go back to our little example here. And remember, overall, if we looked at everybody, there was a significant effect. But if we look at a subpopulation, for example, the green people, if we did the trial just on them, we would not have approved this. And the purple people, well, there's just too few people in this group at all to make any sort of conclusion about should we use this drug on humans or not. And this is a big problem, right? It's big enough that the national academies looked at it. And they came away with the idea that a large number of people in our country do not benefit from medical advances. Do not benefit because we don't represent them in our trials. All right, that's a massive issue, right? And so we're here today, well, what should we do? Anybody? What should we do? Well, we could run our trials slightly differently. We could, instead of saying, oh, let's treat everybody a big group, let's take our groups, let's reshuffle them into sub-trials. This turns out not to work. Like, one, it is super hard to put a person into one group. We all probably straddle multiple groups. So shoehorning people into individual groups is one, hard. Two, well, what do you do if someone that looks different than any of these groups show up? Like, what the hell do you tell them? Like, sorry, we don't have any information for you. So this is not going to work. So what we want to do in my lab is we want to put the patient first, and then we want to use these massive data sets to go through and find other individuals who look like our patient. We want to look at their medical records, and we want to figure out what dosages work for you. In fact, we actually want to give your doctor a device. So sitting with you, they can say, well, 2.5 was supposed to be the starting, but let's find people that look like you in our giant databases, and let's see how they did, and we might get this new result that says, actually, people that look like you, they did much better on 7.5 milligrams. So we're not changing fundamentally how medicine is done. We're giving doctors the data they need. We no longer say, this is how the population responded on average, but this is what might work for your patient better than that initial dosages, all right? So the question is, well, how do I figure out? I said we can't put people into individual groups. So how do I figure out, well, who is similar to my patient? Well, that's a different question, right? Putting people into static groups and then putting a new person into a group, that's hard. It turns out to be much easier to just find who is most similar to you. Now, there are a lot of, oh, sorry, I jumped ahead of my slides. So to talk about genetic similarity, let's talk about the human genome. There are 24 chromosomes. You get one copy from mom, one copy from dad. This is a male configuration. Here's the female configuration. Now, to say that you get one copy from mom and one copy from dad actually hides an incredibly important aspect of genetic diversity that happens in meiosis. So meiosis is that special cell replication stage that leads to sperm and egg. So this starts off like normal cell replication. Individual chromosomes line up. So this happens on all chromosomes at once. We're just going to look at one. The mom copy and the dad copy all duplicate. They move to opposite ends of the cell, and here is where it changes. There's this interesting effect called recombination, where you actually swap genetic material between the chromosomes, and you end up with these never-seen-before mosaics. These mosaics become sperm or egg, and when you have two sperm or egg fertilize, you now have 50% of the genome from mom, 50% from dad, but in a completely unique way. This is why you and your siblings look so different. That rearrangement that occurs in meiosis gives you a unique chromosome. And if we look at the effects of recombination over the generations, we can actually track how you are more similar to mom and dad, less similar to your grandparents, by looking at these long stretches of shared DNA. We actually call these identity by descent regions. You can see I start off with a big blue bar, and that slowly gets smaller and smaller and smaller. And in fact, the cutting edge way to tell how genetically similar two people are is to measure, well, how long is the longest shared segment that you have? How long is the longest identity by descent region? And there are a lot of methods for this, right? This is a really important problem. But the issue with any of these is that they are for finding the distance between any two people. okay? I have millions of people. I can't take a million people, take them into two sets, and then find all pairwise distances between all of them. I end up with this gigantic matrix. But again, I asked this question before, what happens when someone new comes along? Do I just totally start over? So this is not going to work. And it turns out the technology to solve this problem, you may have used already. So Google, we've probably all typed things in. There's another way, it's called reverse image search. You actually take an image, and that image can be your query. So if I take a cute baby giraffe and I reverse image search it, then I will find a bunch of cute baby giraffes. And at no time did I type cute baby giraffe. I just had a complicated object. I searched for other complex objects that looked the same. I think you can see the parallels between what we're trying to do with human genomes and what I'm showing with giraffes. The fundamental technology that enables this is called an embedding. All right, so we're going to kind of get a little bit mathy for a little bit, and I think we all can do it. And I think that if you get a little bit lost, I've got some summary slides to catch everybody up. But after the next 10 minutes or so, I think you'll have a really deep understanding, or certainly deep enough understanding, of how artificial intelligence, machine learning, training, neural networks, all of these buzzwords that have been floating around us for the past 5 years, you'll have a sense of them, okay? So embeddings are lists of numbers that represent an object, okay? So suppose I have some way of creating embeddings. This is magic black box. I take a cute baby giraffe, and what this will do, it'll give me some numbers. And these numbers represent that giraffe. That might seem abstract, so I'm using two numbers here. Think of them as their location on this graph. So I'm putting this cute baby giraffe right there. In practice, we actually use hundreds of numbers for each object, but here two, we can visualize that. So this by itself isn't that meaningful, but what it does, it creates this embedding space, which I have this graph here. If I add new images to this, they will end up landing, if they are similar to each other, they will have similar numbers, and they will be close to each other in this embedding space. The power here is an embedding gives structure to a really large group of complex objects. And the power is, if I have a new image and I want to find similar images, I can get the embedding and go look in that spot. And I find other things that have similar numbers, and if I did all this right, I will have other things that look the same. Make sense? And hopefully you see where I'm going. We're eventually going to do this with genomes. Now the question is, well, where in the hell do you get this black box from? There are a lot of ways to do this. We're going to focus on using a specific type of machine learning called a neural network. And I promise you, these are not scary. They might look scary because this is how people draw them. This is a total tangled mess. But in fact, all this graph does is show instructions for carrying out very small mathematical operations. And we're going to see that with just one path through the graph, okay? We start off with some numerical input, and we're going to every hop here is going to transform that number just a little bit using two values. One's called a weight, and one's called a bias. We're going to take that input, we're going to multiply it by the weight, and then add a bias. And I'm simplifying things a little bit here. But overall, so if I had 0.1 as my input, and these are my weight, this is my weight and my bias, I would end up with 0.2. If I put a weight and bias at every step, I carry that math all the way forward, and I've transformed my input to my output. Okay, well, who cares? Well, the exciting thing about this is I can look at that 0.3, and I can tell, well, is this good or not good? And if it's not good, I can actually update these weights and these biases. So for example, if this 0.3 changes to a 1.3, I run the math again, I now get a very different output. So the whole point of a neural network is, and why it's used for image recognition, for speech recognition, for finding similar genomes, is that you can mess with your weights and biases until you get a really good output. So just a quick summary, if that was too much. We have an input, a number as an input. We modify those by weights and biases. Those are just names we gave to numbers. And it produces an output. And the whole game is to adjust those weights and biases just so we get our intended effect. And that process of changing the weights and biases is called training. And in particular, we're going to talk about supervised training. And this is kind of like a trial by error. So you start off with a bunch of cute baby animals. And the supervised part is that I've labeled what animal is in the picture. So that's all supervised means. I have some input, and I have some information about each input. And I start off with a neural network with totally random weights and values. So what's your guess? If I have random weights and values, what will I get? Yeah, I will get an embedding space that is, sorry. I will get a nonsense embedding space. You would never put a cute baby giraffe next to a cute baby elephant, right? And so this is what happens when you have random. So now I need to go back. I realize this isn't very good. I need to update my weights and my biases in my neural network to make this better. And we do that in a very deliberate way. We take these objects a pair at a time, and we compare their label to their embedding. This label is very similar. The embedding is very far away in space. So what do? Move them together, right? We update the weights and biases, and we slowly move these two things a little bit closer. And we do that for another pair. We see the giraffe and the elephant, and we see that their labels are different. I mean, they're both extremely cute, but they are of different animals. But their embeddings are super close. So what do? We spread them apart. We update the weights and biases so the embeddings are further away. And we do that over and over and over and over, millions of times, and we end up with an embedding space. We end up with a neural network that has weights and biases that are tuned to give us this useful embedding space. Okay? So that's it. You now are trained machine learning engineers. And just a little bit of recap. So embeddings are lists of numbers that tell us something about the object. In particular, they help us compare objects. We used a neural network to learn the embedding function. That's a real power of neural networks is you might spend your entire career trying to write down some sort of formula that would convert an image to some numbers that represent that image. Or you can just take a bunch of examples and let those examples tweak the weights and labels just so until you get your right output. And then in the end, we have numbers, and these numbers are powerful because numbers that are close are similar objects, and numbers that are really far are distant objects. And that will help us find similar things. And this is exactly what we did with Genesis. This is Kristen, my PhD student, who will just submitted her dissertation to her committee. Good job, Kristen. Her and Murad, I don't think Murad is here, worked on this project. It's called Genotype Similarity Search, or Genesis. And we did exactly that. We took two genomes, we fed them into a neural network, and we got the embeddings. We then used one of those existing methods to compare the distance between the genomes and the distance between the embeddings. And we did this a million times until we had consistently were producing embeddings that were as close as the original genomes were. Okay? And to give you a sense of scale, we used 100,000 weights and biases in this. Is that a lot? ChatGPT uses 1.8 trillion. Now, I have problems with orders of magnitude, and scale is difficult for me to imagine. Microsoft, who owns ChatGPT, just purchased 3 Mile Island nuclear facility to power ChatGPT. That is both fascinating and completely terrifying. But that gives you a sense of scale. We did not need to have a nuclear power plant to train our method, but these advanced AI, they do require massive amounts of compute hardware and massive amounts of energy to train and to run. Ours is smaller. All right, so this is what we have. Now the question is, does it work? And so to test whether or not this works, we used data from the 1,000 Genomes Project. Now, if you learn anything, you should learn never name your project with some sort of number, because the 1,000 Genomes Project had, you guessed, 3,202 genomes. You're never going to hit 1,000, so don't even name it that way. So what they did is they intentionally sampled human genomes from around the globe to better understand global diversity. Now, you can have some arguments here that's probably undersampling Africa. Native American tribes excluded themselves, Native American tribes excluded themselves from this, not surprisingly, given how human genetics have treated them. But overall, this is a sampling of human diversity. And what we're going to do to evaluate our method is we're going to leverage the fact that people, so you see these blue dots, those are all Europeans. The upmost dot are Finnish people. So you have two levels of granularity here. You have subpopulations and you have superpopulations. And we're leveraging this fact that since we used to be fairly isolated, we're not isolated anymore, but we used to be, our genetics are such that, for example, Finns are more genetically similar to other Finns than they are other Europeans. Europeans are more genetically similar to each other than they are people from other areas. So we're going to use this geographic constraint as a proxy for genetic similarity, and then we're going to ask, well, can we find, for a given individual from some of these populations, can we use our method to pop back out groups that are mostly people of that matching population without any knowledge of the populations to begin with? Make sense? Okay, so we take all these people, we use our embedding, we find their embeddings, we put those embeddings into a big database, and we search. And from this search, we get back out, here are the individuals who are most similar to my search, and I'm actually going to put them in order. The person who is most similar is first, second, most similar second, all the way to the least similar. As you grow the size of the group you want to look at, you are necessarily going to include less and less and less related individuals. That's just how it works, right? So we've sorted them by the most similar to the least similar. And then we've produced what we call these decay graphs. So with these decay graphs, measure at what speed does the similarity of your cohort drop off. So we look at a cohort of just one. So that's going to include the most similar individual. In this case, that person matched the cohort, so we stay at 100%. And we stay at 100% until we get to this purple person and we drop down. So we are starting to see some decay in the similarity of our cohort. And we do that all the way across, and we get this overall curve that, as I said, tells us how quickly the similarity drops off. And this is interesting when you compare it to something else. So the other best method out there, the pairwise method, that's really good, it's called King Robust. And this is just fake, but I've added one change to their cohort. I've just put a purple person in place of a green person. So one change, different there, and you would see this decay curve. So you can see the decay curve reacts to not only what individuals are outside your population, but how similar did you think they were? So if I pushed that purple person further down, you would see much less of a change. But since they were way further up, you see a big difference. So this is our decay plots, and we're going to look at Genesis versus King Robust. Genesis is going to be a straight line, King Robust is going to be a dashed line. And for European populations, we do quite well. We have a much shallower decay curve than King Robust, and that's true for almost all populations, with the exception of the African population, which is by far the most diverse population out there. Any two Africans are more diverse from each other than they are from any other two people, which is one of our best evidence that humans started in Africa and then left. But yeah, so we do quite well, not only in finding good cohorts, but also across all these different populations. And it's important to look across populations because you want to, like the warfarin example, we don't want to just focus on Europeans and leave other individuals in the dust. All right, so this works pretty well. We also tried it on UC. This is real data. So UC Health has a biobank. Anybody here part of the biobank? Very good. If you're not, you can go on my chart. You can enroll in the biobank, sign some consent forms, and be part of our next version. But right now, there's about 200,000 people who have consented. They're slowly sequencing everyone. We have about 74,000 people. We have their genetics and their medical records. And when I look at, and we can run these, I say, and Kristen, can do this very, very quickly. In less than a second, we can find your matched cohort, which is certainly fast enough to live up to that dream of your doctor having a handheld device, can click a button and get new results specific for you. And for the most part, our decay curves look pretty damn good, right? Except for some populations. For Central, South Asian, and Middle Eastern populations, we have horrible performance. And digging into this, it's because they're actually drastically underrepresented in the database. For example, Middle Eastern individuals are 0.3% of the entire database. So they're basically flooded out by everybody else. So it leads us to the problem of, well, we need to deal with inequities. We're here trying to fix both you getting the right medication that you need, and also trying to make sure that everybody can get the right medication that they need. But are we creating inequities along the way? So we always need to be self-reflective and make sure we're not creating a problem here. And the one that we looked at was called database inequities. What happens when there's no one like you in the database? So I started off with this example of, I'm going to take this green person and I'm going to search them. Well, this is a totally artificial scenario. This database exists solely because people went out and specifically sampled diverse populations. If we're going to use a biobank, well, biobank, who's in the biobank? CU Health patients. Who's a CU Health patient? People in Colorado. So the diversity of the biobank in Colorado is going to match the regional diversity of Colorado. So instead of having this search criteria where you're searching a very diverse population, depending who you are, your search is going to look like this. Either there's going to be a ton of people that look just like you in the database, or none. And so this creates a massive problem. Fixing this is quite hard. Like, how do we increase the diversity of people signing up? Maybe through talks like this. But what we can do is actually, we can take our black box and we can share it with other institutions that have different regional diversity. We can create one big melting pot of this, and it doesn't matter where the patient walks in. If I can't find a representative set in my database, I can go out and look in other databases. You know, for example, a Middle Eastern individual comes in, well, I probably have better luck looking in a Michigan database. Or a South American person, maybe UCLA is better. So it doesn't matter where you come in, if we can distribute this solution across institutions, and we can actually do this without sharing patient-level information. Really exciting. Everybody keeps their own databases, and we just share, oh, people that look like that person, 7.5 milligrams, right? So you share no individual level information, which makes it a lot easier to distribute and create this big network, we call it federated learning. So this big network of machine learning instances that are put out, and we can search across all of them. And we're actually trying to do this. This is not a joke. I led a large grant to ARPA-H, which was a fairly new funding agency, and the whole point was to do exactly what I'm saying. For common diseases like hypertension or type 2 diabetes, we want to create these databases and we want to distribute them to primary care providers. We actually want to push personalized medicine out to the edges. So we can have start with you, take a large population of individuals, use machine learning to find people that look just like you, and from that, we can answer questions that you want and your doctor want answered, like, what should I take for hypertension? How can I control my blood sugar? There's a bunch of options. Metformin, what should I do? And then I think you can imagine, well, what chemos would make me sick but not actually shrink my tumor? What is the point of going through that pain if it's not going to help you and make you really sick? All of this information is encoded in the health record. We just need to find those people that look like you and look across them to see what works best. And from that, we can then update your physician. All right. So yeah, that's it. I always show a picture of all my kids at the end, and I'm happy to take any questions.